presentation. So thank you everyone for joining today's webinar. This is the second webinar in this series that we've been hosting to learn more about machine learning. And today we'll be unlocking novel insights, novel spatial insights with graph neural networks. So this was set up in our previous introduction to machine learning that was covered by Amaro on January 25th. And to reiterate, the goal of this webinar series is really to empower our partners with our AI approaches in digital pathology and its applications for innovation and development. If you have any recommendations of how we can improve this webinar, if there's topics that you're interested in, please put them in the chat or email us afterwards, and we'd be happy to consider them for future material. Can we go to the next slide? So just to put this out there, I'll give everyone a moment to read this, but this is just basically to inform you that this information is for a general audience and it's not meant, should not be construed for professional or medical advice from PathAI. So I'll just pause here for a moment, let you read through this. All right, next slide. So as I mentioned, today we'll be covering unlocking novel spatial insights with graph neural networks. And before I begin, I would like to introduce today's speakers, which are Robert Egger, who's a biomedical data scientist here at PathAI, and Michael Drage, who's the director of pathology here at PathAI. For those of you who don't know me from the previous webinar or haven't met me in the past, my name is Rob Brocken. I'm the director of biopharma partnerships here at PathAI. Next slide. So today we'll be covering challenges in machine learning for digital pathology. These were discussed last time by Amaro. I'll just quickly review them. And then we'll dive into the overview of graph neural networks and providing examples that will help outline the approaches with those networks. And then we'll have a Q&A session at the end. To remind those of you who are new to this webinar series, please post your questions into the chat. I'll be monitoring them and collecting them, and we will have a Q&A session at the end with Robert and Michael. So challenges in machine learning for digital pathology. The areas that we identified last time were interpretability and explainability. So obviously, the ability for a pathologist, clinician, or patient trust is required in this context. And also it's required for generation of hypotheses and scientific utility. We also have to consider overcoming the technical covariates. So this is variation across samples, scanning and staining those pre-analytical variables that may be introduced as part of this, and how to identify and isolate non-spurious signals, and then considerations around data security, data scarcity and curation. This would be the construction of representative data sets with limited sample channels, and then limited and non-uniform metadata. Next slide. So when we're considering algorithm development, obviously keeping in mind your end goal of what you're trying to achieve with this model is always important. But you also have to consider the data, both the breadth and depth, to minimize the bias and errors with while maximizing that predictive utility that you're seeking. In terms of the depth, could be the orthogonal data sets, the real world data, outcomes, genomics, or any other associated data set that you may have. And then the breadth of the data. So the multiple scanners, stain types, multiple labs, multiple indications. Again, considering all of the pre-analytical variables that your model may have to churn through to be robust enough to deploy in the context that you're trying to use it in. Then considering the model itself, what is the best approach for this? So is it a predictive model? Are we just going to be using an end-to-end approach? Is there a spatial approach needed? Do we need the biological insights? Is it a generative model? All of these things need to be considered when developing these AI tactics. Today, obviously, we'll be covering the GNN approach. And then the evaluation. So the rigorous quantitative evaluation principles based on model approach plus the context. So what is that context of use? Is it RUO? Is it for a prospective study? Is it going to be used as a DDT? Or could it be a potential diagnostic? And then looking through the evaluation type. So we use consensus here a lot, but there may be a different evaluation modality that you may be interested in. So back to the models. Last time, Amaro gave us an introduction to convolutional neural network segmentation models. Today we'll be covering the GNNs, or the graph neural networks, and then we also will have the end-to-end models and generative models further down the line. So with that, I'll kick it over to Robert to cover GNNs.
Thanks, Rob. Yeah, we'll be talking now in the first part, we're going to give a brief overview over why spatial modeling for biomedical research, digital pathology, what GNNs are. And in the second part, like Rob mentioned, we're going to go through some examples in more detail. But first, I would actually like to draw on Mike's experience doing biomedical research to give us a little bit of an intro why spatial modeling might be particularly relevant here.
Absolutely. So I think probably it's worth reviewing just a very fundamental concept that I think is intuitive to everybody on this call, but it's worth calling out in this particular use case because in GNNs, because it's highly pertinent to GNNs specifically. So in general, the fundamental concept is just that structure in biology, structure always relates to function. And so if you look, if you think of, well, if you look at this left panel, you have early stage fatty liver disease in the upper panel and late stage like cirrhosis, established cirrhosis and fatty liver disease in the lower panel. And you can see that there's been this extensive remodeling of the liver architecture, both in terms of the mesenchymal component, which was previously, limited to just the portal tracts that developed during development, to now this, extensive fibrosis that is bridging between different portal tracts and central veins, what used to be central veins. And you have this, and the lobules are also totally reorganized in terms of their, the zonality is altered and in many instances lost. And you have this like loss of this zonal definition of functions of hepatocytes as well. And so you have this tremendous like alteration. You have all this biliary proliferation, you have the vessels. So I guess to compare it to the previous like 2 slides when we were talking about different model approaches, like you could capture something like the area proportion of fibrosis, at the slide level, but you wouldn't be capturing the sort of the relative proportions of the vessels and the biliary epithelium and the fibroblasts without using a GNN that captures this spatial rearrangement, which I think is more informative. And you can find a similar, on the right, this is referencing a citation from the literature, where spatial analyses have also been useful to look at the relative approximation of cell types to each other. And so in this case, it's a score that is based on the relative proportion of benign CD4 T cells to tumor cutaneous T cell lymphoma cells and T regulatory cells, and how it predicts response to pembrolizumab. It's a little bit different. That's not a GNN. That's a cellular neighborhood analysis, and I think I'll hand it off to Robert to discuss the differences between what the potential advantages are we see in GNNs.
Thanks, Mike. So basically, what we're trying to do here is to take this like very basic principle for biomedical research and explicitly use spatial information directly when modeling, for example, disease, or when trying to uncover new spatial signatures in the data. And while we get into that, I want to start with like a little bit of terminology just to clarify what we're all talking about when we say graph neural networks. What does that mean? So let's start with the first part, graphs. What are graphs? Graphs are are mathematical objects. They contain what's called nodes and edges. And they can be, these mathematical objects can be used to represent such spatial biological data like fairly well. For example, you can interpret cells in tissue as the individual nodes. So this is an example on the left in that kind of picture. Each dot that you see there in different colors is a cell. The color, the location is the location of the cell in the tissue, and the color is the cell type as determined using multiplex immunofluorescence in this case. So one approach is to simply assign each cell as a node in the graph, and the edges between nodes can then be used to describe the spatial organization, in this case, of the cells in the tissue. So you could, describe the cells in this tissue by a graph where nearby cells have edges formed between them. And that basically implicitly captures the entire spatial and topological organization of the tissue. And so now we have a graph. So what's a graph neural network? Well, as the name says, it's simply a neural network that operates on a graph. What does that mean? It's a neural network that, in contrast to commonly used convolutional neural networks for images, it simply takes in a graph and is then able to learn and automatically identify patterns in the data that are relevant for the task at hand. So let's go a little bit to the for how we think GNNs can be useful for biomedical research. We want to go back to Mike's comment and really say, what's really important is spatial information because it intimately relates to function. So the way we approach this is we look at model predictions on images. So in the left panel, you see in color the segmentation result, for example, from a CNN that labels all cells and the background tissue in this little snippet of an image. And different colors might represent different cell classes, for example. So what do you now do with this information? So this is a very small part. In general, we work with whole slide images, as are common in digital pathology. So those contain thousands to even millions of cells. So what do you now do with this information? One approach that we also use very successfully is to define features of interest that can then be computed over the entire slide and summarized at a global level. For example, you can measure the relative abundance of tumor-infiltrating lymphocytes. That's something that you can, we know that those are an important feature for immunotherapy. So you might be interested in what's the relative abundance of tumor-infiltrating lymphocytes in a slide. Now, the disadvantage of that is it really relies on you already knowing which features are important. And, we might not always be aware of what kind of subtle spatial features are indeed related to whatever quantity or outcomes we're interested in. So this is where GNNs, graph neural networks, now come in. Graph neural networks are able to automatically identify subtle features in the data, and they don't have to be on a global scale. They can be like really localized patterns even, and identify which of these patterns actually associate with outcomes without needing to pre-specify these features. So it's a much more flexible approach, if you want, compared to pre-specifying features a priori. And one thing that's important to note here is, again, we typically apply these graph neural networks on top of the model predictions that come out of, for example, convolutional networks. So these GNNs work on biologically relevant objects such as cells or tissue. That means an additional advantage of using GNNs is they're highly interpretable. So the input that goes into these neural networks, those are biological entities where we, kind of have an understanding of what they're good for. We know that cells are the elementary constituents of tissue. We know what different cell types may do. So when we later on, we can go back and interrogate these graph neural networks to identify, maybe there's only a certain subset of cell types that are actually related, that the neural network identifies to be related to predictions of interest. Okay, so how does this look in practice at PathAI? How do we train these neural networks? Let's walk through this pipeline a little bit step by step at an abstract level first, on the example of how we could create neural networks to predict some orthogonal metadata. And we're going to go into a more specific example right after that. So I'm going to walk you through this pipeline from left to right. So the input to graph neural network, as I just mentioned, these are, the first step is to extract biologically meaningful objects that could be cells or tissue areas. The next step is to now aggregate this information, so these biological objects, at a relevant spatial scale. That depends a little bit on the problem at hand. That could be like individual cells. We said, you know, individual cells could form the nodes in a graph. It could be groups of cells, or it could be tissue structures. And so those now form the nodes in a graph. And now these nodes carry quantitative features with them. So as an example, if you're looking at the cellular level, that might be information about which cell type is each cell, how large is each cell. Or if you're looking at groups of cells, you could ask, like, you know, how many cells are in this cluster versus in an adjacent cluster, and which cell type do they belong to. In the next step, we incorporate the spatial organization of the tissue by forming edges between nodes that are nearby. And finally, we take these graphs. So we take all the data that we have in our, you know, training data set, move them through this pipeline so that every data point is now represented as a graph. And we now train a GNN to associate features in this graph, features and spatial substructures in this graph with the relevant task variables that we're trying to predict here. So from the rather abstract pipeline, let's go into like a more specific example for how to actually do that. So there's broadly at PathAI, we have three categories where we apply GNNs. The first one is improving whole slide scoring using spatial modeling. It's going to be the first example. The second one is identifying spatial signatures of known genetic biomarkers. This is something that we're not going to talk in so much depth about, but we have a poster of that on our website. And the third one, we're going to talk in more depth as well. That's really to use GNNs as a discovery tool to identify novel spatial signatures and generate new hypotheses for biomedical research. So let's start with whole slide scoring, which mostly closely resembles the pipeline that I just introduced. And I would appreciate if Mike could give like a brief introduction to the example that we're going to cover here, which is NASH scoring.
Thank you. Yes, so when pathologists review liver biopsies for fatty liver disease, we score the activity and stage of the disease using a very standardized criteria that are called the NASH scoring system. So when we quantify semi-quantitatively the ballooning, the steatosis, and the lobular inflammation. And then we also semi-quantitatively evaluate the fibrosis score to put it into a 4-tier staging system. It's well known now that there's a surprising degree of variability amongst pathologists in basically all of these aspects, probably the least of which is steatosis, but in terms of lobular inflammation, ballooning, and fibrosis staging, there's certainly considerable variability between expert pathologists, even amongst the pathologists who worked as a group to establish the staging criteria.
Thanks, Mike. So it would be great if we can use spatial modeling to, you know, create a tool that helps us reduce that variability a little bit. And let's walk through how we went about developing such a model. So as Mike just pointed out, there's different components to evaluating NASH on a liver biopsy, and this is performed on usually at least two pieces of tissue, one of which is stained using H&E, and the other one is stained using Mason's trichrome to reveal collagen and assess fibrosis. So, and in line with the pipeline that I just described to you, we've developed a GNN for the H&E based scoring process. This the first part of the pipeline is to take the actual H&E whole slide image of a biopsy and segment out areas of steatosis, ballooning, and lobular inflammation that are color-coded here on that same example image. We then turn these segmentation results into a graph representing this H&E biopsy and train a graph neural network to predict these NAS activity score. And the training data is on one hand these H&E images with where we segment out these different tissue areas. On the other hand, we have a set of labels provided by expert annotators that tell the model what the value for each score should be for this piece of tissue. And we have a parallel pipeline for assessing fibrosis from Mason's trichrome stained slide. In the first step, we segment out the fibrotic areas. We turn this tissue segmentation into a graph, and then train another graph neural network to relate these segmentation results to the fibrosis scoring provided by an expert annotator. And so this is an iterative process, as all neural network training. So you make a prediction, you compare it to the ground truth or expert annotation, you measure what the error is, and update the neural network as a result. So this iterative process is repeated until the network until the network performance does not improve anymore. And now the question is, did we achieve our result of reducing scoring variability? To assess that, we looked at model performance on a held out test set. And let me take you through this graph. So here we're assessing performance using Cohen's kappa. And here in this table, each row is one component of the NAS score and the fibrosis scoring. On the left, you have no agreement, so here kappa would be 0. On the far right, perfect agreement, kappa would be 1. The black circles are, as a reference, the inter-annotator agreements. So if you look at individual pathologists and compare how they assess individual cases, those are the values that you would get here. And as Mike pointed out in the beginning, this is something that, depending on the score, for steatosis, they're fairly consistent. Other parts of the scoring system are more difficult to assess in a consistent manner. And in purple, you see the comparison of this GNN that we just described in terms of how it compares to the consensus. So the agreement between the GNN and the pathologist consensus is higher than the agreement between pathologists amongst each other. So this is an application where we're able to use GNNs and the spatial modeling at the tissue level to improve reproducibility and reliability of this whole slide level scoring. So let's go to the next application, discovering novel spatial signatures. And here, this was a collaborative project with scientists from Bristol Myers Squibb and Akoya, where we collected a data set using the Akoya PhenoImager. And for those of you on the call who are maybe not familiar with multiplex immunofluorescence, maybe, Mike, can you give us a little bit of an intro of what that technology is, why it's useful, and maybe what some of the limitations are that we would hope to overcome with GNNs?
Absolutely. So the field of tumor immunology is really dependent on precise immunophenotyping of the immune system cells present in the tumor microenvironment. So it goes a little bit beyond what you can do with an H&E image. So for instance, you can do a lot with an H&E image, and you can learn a lot about, how tumor infiltrating lymphocytes are important for immune checkpoint blockade. But you can go much further if you use multiplex modalities where you can quantify, amongst not just lymphocytes, but you can tell that they're T cells, you can tell that they're CD4 or CD8 T cells, you can tell that CD8 T cells are cytotoxic or if they're in an exhaustive state. So you can get at more functional, more functionally relevant data that is, more granular and more specific to, as a stronger input onto predicting immune response and understanding the biology of what's going on. So I think that's where, like, the more data you can put into your machine learning approach, the better it's going to be.
Right, and of course, like what everyone's after is discovering novel spatial signatures that go beyond hypotheses that we've generated thus far based on tumor-infiltrating lymphocytes. What else is there, basically, right?
And I guess I would add, just as a really quick thing, sorry, is that the more complicated the phenotyping gets, the more difficult it is to manually observe the differences, right? So if you're starting to think about, maybe like 12 different cell types, and you're trying to evaluate the spatial relationships between these cell types manually, that's basically impossible. Like, you're not going to be able to do this amongst hundreds of cases and get a signal that actually makes sense. So I think that's where we really need machine learning models to step in to get that signal out, to extract it.
Right, and nowadays people go up to like 40 markers in codex, or like hundreds of transcripts when looking at spatial transcriptomics, for example. So it basically becomes unfeasible to look at pairwise relationships, right? So let's talk about how we tackled that problem in a scalable way using GNNs. So the data set that we worked with here was put together and imaged by Bristol Myers Squibb. And where we came in was after segmenting these multiplex IF data. That's like it's a common step where where the field is, we're getting really good at like segmenting these cells out. Where are they located? What kind of phenotypes are they? But what do we do now, right? That's where we came in with our graph neural network. So on the left, you see the result of this phenotyping approach. So in the panel on the left, each dot is the location of a cell, and the color of each dot is the cell type according to the phenotyping that comes out of this multiplex IF and cell segmentation pipeline. So in red, we have cancer cells. In orange, we have fibroblasts. And the other colors reflect different immune cells. And as you pointed out, Mike, for example, we have our different T cell subtypes in there as well. So what we did here was we didn't tell the GNN to look for a specific target. Rather, we used the GNN to, in an unsupervised fashion, look for patterns that occur repeatedly across this cohort. We had about 150 cases here. And what the GNN is able to do is to look at all these markers at the same time. So each node in the graph that we had here was a cell. It had information about what cell type it was, how large the cell was, and it had information in terms of the graph topology, what cell types are immediately surrounding to it, maybe a little bit further away. And the GNN is able to identify which patterns defined by both the spatial organization and the protein expression of these cells occur repeatedly across this data set. And that's what you see in the third panel. So here we found that this data set is well characterized at a high level by 3 what we call spatial phenotypes. So each cell in this data set falls into one of these 3 buckets. Blue, green, and orange. And, because we can always go back to this is the nice thing about these, the interpretability of these GNNs, you can go back to the input data, which are, cells. So first of all, you can compare the third panel to the first panel, and you see that the green cells are mostly located in the cancer epithelium. It's a lot of cancer cells, but it's also some immune cells in there. The blue cells and the orange cells are more located where you find a lot of fibroblasts, but they're not exclusively stromal cells. There's also some tumor cells in there. So it appears that the model learned something that there's like some solid tumor areas, but there's also these areas where the cancer stroma cancer and stromal interface, that's like a feature that we didn't put into the model at all. We know it's relevant for immunotherapy, and this is something that the model is kind of like able to identify itself. That's like a repeating pattern in this data set. So what's the difference between the blue and orange spatial phenotypes? Well, one thing that you're also able to do, because this is, the input to this GNN is individual cells, you can do like a simple enrichment analysis, for example. So that's the panel on the far right. Each row in this matrix is one of the spatial phenotypes, blue or orange or green. And the column is the relative abundance of the different cell phenotypes. So in the green spatial phenotype, cancer cells are enriched while other cell types are underrepresented, but not zero, but underrepresented. In the blue and orange phenotypes, spatial phenotypes, fibroblasts are enriched, consistent with this interpretation that we're looking at the, cancer stroma or cancer stromal interface. And the difference between those two spatial phenotypes is the relative abundance of different immune cell types. So on the orange spatial phenotype, these CD8 and FOXP3 positive lymphocytes, as well as CD68 positive macrophages, are enriched relative to the blue spatial phenotypes. So this is like an immune enriched phenotype versus the immune low phenotype. So this is neat, but we wanted to know if these kind of spatial phenotypes are also biologically interpretable and carry, interesting, relevant information about the tumor biology here. So let me walk you through this slide because it's very dense. So the first thing we did was ask, do we see any regularity in these spatial phenotypes across our data set? So one thing that's very simple to do is simply for each case in our data set, you can ask, how much of each phenotype is present in the data that we have. So you can, of the, let's say you have an image that has 1 million slides, 50,000 are in the blue phenotype, 50,000 are in the green phenotype, and the remaining 900,000 are in the orange phenotype. So you do this for all the data that we have in our data set, and you ask, do you see any kind of like systematic patterns emerging? And in fact, we did. We're able to, again, using unsupervised methods, cluster our data set into 3 groups that differ in their spatial phenotype composition. So this is these type 1, type 2, and type 3. Type 1 is enriched in the green immune low tumor phenotype. Type 2 is highly enriched in the immune-rich stroma, cancer stroma interface phenotype. And type 3 is more of a mixed group of cases. They have like roughly equal abundance of these 3 spatial phenotypes. So this is nice. So there's like really some regularity at the if you look at an entire case and not only at the cell level. And moreover, what we were able to do here is we had complementary data available from the same tissue blocks. We were able to use bulk RNA sequencing to derive some RNA-seq signatures. In this case, an MHC1 antigen presentation pathway signature and an CD8 immune exclusion signature. So those are the 2 panels on the right. And the first thing that I want you to note is you can probably appreciate that they're different between these GNN-derived stratification. And let me kind of like, there's a lot going on here. So I simplified these kind of bar graphs a little bit into a little bit simplified cartoonish table to understand what's going on here. So this table is again on the left, beneath those 3 case groups that we derived from using the GNN. In the first type, we see that the antigen presentation pathway is downregulated. So let's say for simplicity, it's not really active. On the other hand, the immune signature is kind of like a mixed bag. It's a little bit all over the place in the right panel. Type 2, we see that the antigen presentation pathway is up, so it seems to be functioning at least normally. Immune activity is also, there seems to be immune activity in the tumor as well. So we put two check marks there. For type 3, we see that the antigen presentation pathway is also active. However, there's a lack of immune activity in these tumors. So this is like pretty neat. This is nothing that we put explicitly into this model, especially something like, antigen presentation pathways. That's like intracellular mechanisms. And yet there is an effect on the spatial organization of the tissue that this GNN is able to identify as a prominent feature in this data set without us telling us anything about it. So these GNN-derived phenotypes and the resulting data set stratification we're able to relate it back to kind of like, well understood biology describing the interaction between the tumor cells and the immune system. Finally, we can also look at whether this all results in whether this has prognostic value, and indeed it does. So if you look at the GNN-based stratification, you see that this group 2 that is enriched in this immune-rich stroma and has both the antigen presentation pathway and the immune activity, these cases have favorable prognosis compared to the rest of the cohort. And I think what's really interesting and kind of like an important question is, after all of this effort, is that really something that's necessary, right? I mean, yeah, maybe it's difficult to look at, pairwise combinations of different markers, but it's not impossible when we're only looking at 6 markers like we do here at least. So we were able to do a control analysis where we said, okay, let's look at the same data set, but not go through this effort of, doing this very complex spatial modeling using a GNN. What happens then, you can do this in many different ways. We've tried a bunch of them, and you're not able to identify these spatial patterns unless you use a GNN. That's one example of that is shown on the right. You can similarly derive like an unsupervised stratification of these cases using other methods, for example, using nearest neighbor analyses. And you arrive at different kinds of stratifications of this cohort that doesn't as neatly relate to these gene expression patterns that we identified on the previous slide. It also has no prognostic value. So this is really a unique view into this data set that you get only when using GNNs. Okay, so let's summarize a little bit what we talked about. Talked a little bit about why we think that GNNs are a great approach to model the spatial context and spatial relationships between cells and in tissue. Importantly, compared to many other neural networks used in digital pathology and biomedical research, these networks don't operate on images directly. They operate on biological objects. Another neat advantage, in addition to the interpretability that we described a little bit, is in principle, these GNNs are more or less agnostic to the kind of like data source, right? We showed you an example from using H&E and Mason's trichrome. We showed you an example using multiplex IF. You can in principle scale this up to other methods as well, like, MERFISH, other spatial transcriptomic methods, as long as you have biologically relevant objects that you identify using some other method. And we went through some applications from whole slide scoring to like really discovery-driven research. Some implications are, using this unsupervised GNN approach, this now really enables us to achieve this goal of not having to pre-specify what it is that we're looking for. We can really look at the complexity of the tissue at the same time, using a couple, 10 markers, 50 markers more, and really understand without any preconceived notions or hypotheses, what are the actual patterns that we identify, that we see here in the tissue. yeah, so, and this is like a pipeline that's plug and play, ready to go. So any kind of data that we get here, we're able to rapidly experiment with and get completely orthogonal kinds of insight relative to other data from this approach. With that, I'm going to end it here, and we're open for questions.
Thanks, Robert. We did receive a few questions throughout the presentation. If anybody has additional questions, please just pop them into the Q&A chat there, and we'll be sure to answer them. One of the first questions is, how many ROIs images do you have to train your GNN to get reliable results?
Yeah, that's a great question. I think in one of the slides we wrote a rough guideline. Going to say what no one wants to hear. It depends a little bit. So the guideline we gave there is depending on what kind of setting you're going for. If you're going like in the setting that I just described, this unsupervised, if you have cell level data, talking about that millions of cells, this kind of unsupervised approach, that can work in as little as around 50 samples. If you have like, if you're looking really at the cell level in those samples. If you're looking more at improving like whole slide scoring, then you probably want to start with, you need like several hundred samples to start with to get something relatively robust to work there.
Great. Another question was, can we use each individual cell nucleus as a node and then analyze the proximity or spatial organization of nuclei in a cluster of cells of the same type?
Yeah, I think that's also something that's possible in principle. That's very similar to the approach that I just described with this multiplex IF data. And now you would be looking in cells of the same type. So yeah, so that would really be focusing on the relative spatial organization of these cells. But yeah, I think that would work equally well. Yes.
Okay. One here is, what is your experience of using GNN models to analyze a series of Z-stack images for 3D spatial organization prediction?
Yeah, I would love to do that. We generally, the kind of data that we work with is really mostly 2D data. So, in digital pathology, the standard really is thin sections like 4 or 5 microns. That's what's typically done for classic H&E trichrome staining. That's also what's typically done for multiplex IHC. I would love to see how this works in 3D. I think it generally, like I said, these GNNs, they're relatively agnostic to the data source. If you can model the spatial relationships in 2D, I don't see why you wouldn't be able to model it in 3D as well. That would be, if it's more a question of who has data sets, large enough data sets to analyze in 3D. But yeah, in principle, I think this is a great tool.
Okay. Another question here, have you tried GNNs for cell phenotyping purposes?
Yeah, that's a cool thought. You could, something that you can do in GNNs is what's called a node predictions. So you can say, I don't know, I build up my data set. I don't know what this node in the center is. So you could imagine, you have difficulty phenotyping individual cell in such multiplex IF data, for example. But what you're able to do now is if you have enough data, you're able to learn, based on the fluorescence intensity, for example, that individual cells have in a data set and what other cells they're surrounded by. You're able to, given enough training data, you would be able to infer not only based on the intensity, but interestingly, but also based on the spatial neighborhood, what phenotype an individual cell should be. Yeah.
Great. Another question here, can we use trained GNNs across similar data sets? So I think this is more relation to translation or deployment in additional data.
Right. That's an excellent question. That's something this is where Rob mentioned this briefly in the introduction. One challenge in digital pathology machine learning here is always pre-analytical variables. So for example, if you wanted to deploy such a Nash scoring model on a new data set, the main challenge is to ask whether the segmentation model, for example, is robust against staining variations, scanning variations. We have experience in building models that are that handle some of these differences, but I think that's the major concern. Like some of these pre-analytical variables. At the same time, there's other concerns in terms of does your during training, do the training data reflect, for example, in terms of disease severity, what you're seeing in that new data set that you want to deploy your model on? So you have those kinds of concerns as well. Yeah, that's what I would think about before deploying a model on a completely new data set.
Okay. Another question. We got a bunch here, so I'm going to try to rip through them. One is, when we aggregate the cell information in a local region to form a node, how would you define the region size? The region size might affect the node profile.
Yes, that's an excellent question. Totally, the region size might affect the node profile. And this is really where it's great that that we're working with biological objects here. Depending on the question that you're interested in, this may, even for the same data set, differ. So maybe you care about the individual cells, maybe they're not important and you want to aggregate them at different sizes. In the past, we've done this using two methods. Either we have some, biological information that allows us to constrain how large we should build these nodes. Or if we don't, really, it's an empirical question. And you can almost think of it as a parameter during model development. You might want to try smaller nodes, you might want to try larger nodes if you don't have any other information that would constrain how large those nodes should be.
This, I think, kind of slots into it. Can we define spatial features at different spatial sizes and scales? I guess this kind of would filter into the size of the node, but
yeah, it would filter into the size of node. One other technical note is that these GNNs, the way we build them, they're able to some extent, to identify, if you have smaller nodes, they're able to identify at what spatial scale it is more appropriate to look. So while there's some degree of flexibility, there's also some slack built into the model that it's able to learn, oh, maybe the nodes are a little bit too small, or maybe spatial organization at really long scales is important. And so the models are somewhat forgiving against that, and are able to figure out some of that by themselves using the methods that we've built into them.
Great. One question here is, have you tried identifying cell types in unstained specimens? I think this may be a question that we could point to the April webinar on QMAI and some of the advanced imaging modalities that we're going to be covering there. Do you or Mike have any follow-ups in terms of identifying sample or utilizing these approaches, or any analysis approaches in unstained samples?
Yeah, I think you're right, Rob. That's some of the imaging techniques that we have are more appropriate for that, because, as a reminder, again, the way we typically use GNNs are not based on the images directly, but on information that you get out of these images, for example, from segmentation of multiplex IF data or H&E data, and we then operate on these biological objects directly.
Here's another one. Are the pre-trained models available, or each end user has to train the model themselves?
Rob, here is where you correct me, but I think we do have a set of models available.
Yeah. So PathAI, we provide deployment of our model library that we have. So there are avenues and potential ML stacks and things that could be available to end users, but typically we would train the model for you and deploy it when needed. So once you have a data set. So the answer is yes, there are pre-trained models for specific instances, but it would just depend on the use case for what we would deploy it and if it's available.
Right. And it's also often the case that even if we have a pre-trained model, for example, for tissue and cell segmentation, then you might still want to train a customized GNN for a specific task at hand.
Another one here is, are the input images used to train GNNs processed and cleaned using any classical computer vision algorithms, or we can train GNNs using the images directly?
Yeah, so we typically process the images using like tissue or cell segmentation models first, where, you know, where we do some of this stain normalization, for example. And in principle, if you look in the literature, people have trained GNNs in the past using image information directly. So that is possible in principle. We don't typically do that, mainly because what we're really looking a lot to for these GNNs is this interpretability in terms of like providing really the biological input. When we tend to use multiple instance learning models to operate directly on images. And I think we have another webinar coming up specifically on those and how we look at interpretability in those cases.
Yeah. Quick question here was around availability of the recording. You will have access. We'll be sending the recording out following this meeting when it's available. Another question here is, how many features/nodes are required for training a GNN? Are there any limitations to what needs to be an input?
Good point. So if I want to go with one specific example, let's, for example, this multiplex AF data that I just talked about. Here this was a fairly large data set, thanks to the Akoya PhenoImager. We're able to generate, I think, 150 or 160 images at whole slide image scale. So those were in total 40 million cells. So it's a rather large data set. I've also done this on smaller, it was like, at the cell level, 5 million cells or something like that, which was 20 or 40 slides. So something in that order of magnitude is what you want to do. And in terms of number of features, again, in principle, that's like, we looked at six phenotypes, but this is something that where it actually scales very favorably. And instead of 6, you could just use 100, 200, and the infrastructure works in exactly the same way.
Great. Is it possible, or do you typically run these models or develop these models in a single indication, or do you can this work across indications?
In principle, it can work across indications. I would argue the question is, what are you looking for, right? The examples I showed here were specifically within indication. For NASH scoring, it only makes sense to look at liver biopsies. For for the cancer example, it might be super interesting to look actually across indications and see if there are any regularities that we find that are common across cancers. Although, a lot of things are very specific to individual indications. So it's rather a question of can you build up a large enough data set to do that robustly?
Maybe taking this question to Mike as well. Do you believe that, I guess this is an assumption, but the spatial morphology in a specific indication may just be too great to overcome to transfer? I guess in terms of, if we find this immune phenotype that you kind of outlined in the fluorescence, and we try to apply that to a different, morphologically very different indication, do you think that would be too big of a hurdle, or do you have any thoughts around that?
I mean, I think it's an interesting question, and really it would have to be answered empirically. But I do think there are certain scenarios where I would expect it to be difficult to incorporate different indications, but you could sort of imagine that, core features that are relevant across indications for immuno-oncology interventions might be common, and so you might be able to derive like a more global predictor.
Yeah. Another question is, how does the inference pipeline look for GNNs on whole slide level predictions?
Right. I just go back through that because the Nash scoring is actually a great example of that inference pipeline. So this is essentially the training pipeline, and the inference pipeline essentially looks the same. So what happens during inference is you provide a whole slide image, we deploy the tissue and cell segmentation model, we turn the tissue and cell segmentation into graphs, we then take the GNN that we previously trained, input the graph into the GNN, and make the prediction. So this score now doesn't come from the annotator, from the human anymore, but that's now the output of the GNN. So that's what this inference pipeline looks like.
Okay. To follow up on the previous multi-indication, I think there was a comment here that cross-indication specifically for cancer may be best evaluated in the context of approved pan-cancer therapies, e.g. IO. So, I think that's kind of where that question may have been coming from, as well as, especially if we're talking about metastatic lesions or things where that environment may be holding up in context that that IO, that immune landscape is highly relevant to the specific therapy. But any other follow-up feedback on that, or just a thumbs up? Yeah, that's.
I completely agree. Yeah, that's kind of what I was referring to with like the core features. I think that could be pan indication. But if you wanted to get at like novel hypotheses that might be pertinent to a given indication, then I think you'd want to be focusing on that indication to unveil something that you don't know about.
All right. One here that came in, well, this will be the last question. Do you support open access research? Especially, is the code of this work on GNNs publicly available?
We don't have the code publicly available. What we do have is that we have the poster, for example, describing our multiplex IF work. That's on our website, and there you can look at the pertinent references for the GNNs. They're all in there.
Great. Can you go to the next slide, please? So that was great engagement. I really appreciate everyone for showing up. That was a great list of questions. I'm happy that everyone's thinking so deeply about this, and hopefully is thinking about its application in their own research or development. So just to highlight what's coming next, obviously we have in March, the specific date is not outlined yet, but will be best practices in performance testing and validation, something that is relevant to all of the activities we've been discussing here. And then in April, we'll be going into utilizing machine learning for imaging. So this is that quantitative multimodal anisotropic, I'm always going to mess that up, imaging, a QMAI imaging. This would feed into some of those questions around unstained slides and some of the advanced features that we can extract from even already stained slides and add additional detail. And then in April or May, we'll be building more interpretive and explainable end-to-end models and additive multiple instance learning. So please, again, if you have any feedback on this webinar, let us know. If there's any topics that are burning in your mind that you think we should highlight, then please bring them to our attention. Otherwise, we will see you hopefully in March. Thank you, everyone.